All right. So we know that changes in 3D genome organization is important. So we would like to be able to identify these in a systematic way. So for that, what are some of the, so let's think about, oh wait. Let's think about some of the questions that we might want to consider when thinking about dynamics of 3D genome organization. So the first question is, what is the unit of change? And this can be loops, these can be tags, these can be compartments. And then we want to find these units of change, and then we want to compare them across multiple conditions. So this question of finding differences has been looked at by different methods. And so there are a lot of methods that are out there that have been used to try to find differences at the loop level, so pairs of loci. But not a lot has been done at the higher order level, so at the level of tags. And here I'm listing some of the methods that have been used to try to compare topologically associating domains. And the problem with more, well, I wouldn't say, some of the limitations that you would like to overcome is that they are pairwise in nature. So they don't account for maybe a time course or 12 conditions and so on. So how do we get to that problem? So to address that, together with my student, we developed an approach called TGIF, which stands for Tree-Guided Integrated Matrix Factorization. And this is based on a few key points. It is based on a dimensionality reduction technique called non-negative matrix factorization. And it also leverages multitask learning that I'll get into these in the subsequent slides. It gives you both the ability to look at dynamics at the level of compartments as well as at the level of topologically associating domains. So for those of you who may not have encountered NMF, NMF is a very popular dimensionality reduction technique, and it is very interpretable. So the outputs are immediately easy to understand. And what it does is it works with non-negative data. It has been used a lot in genomics, including single-cell genomics, and also beyond genomics with imaging, text, video, all different types of data. And what it does is it takes a matrix and gives you two low-dimensional representations such that when we multiply them together, we get out as good a representation as possible. But the important thing is that these U and the V, which are the low-rank matrices, are also non-negative, but they also can be used to extract meaningful patterns in the data. So just to give you an example, let's consider the famous Netflix challenge, where we have movies by people, matrix where each entry corresponds to a movie rating, and and if there is a low dimensional structure, so let's suppose that the two types of people, the green people and the pink people, and they like to watch these green movies and pink type of movies, NMF will be able to actually give you this kind of low dimensional representation. Now, we want to use NMF in a multitask setting. So what might that look like? So taking that example a little bit further, so let's say that not only do you have Netflix, but you also have Hulu and you have YouTube. So Hulu and Netflix are both video streaming platforms that give you similar kind of content. But you also have YouTube where you might watch some things. But it is not exactly the same because you also have educational videos, which could be there on Netflix, but I haven't seen it. So in general, Hulu and Netflix are more similar, but the other one is different. But there is this general common thing where there are people watching things, and we would like to be maybe incorporate all of this information together and incorporate that to be able to get more meaningful and maybe more constrained low dimensional representation. And we do that by extending the overall objective of NMF by incorporating this regularization term. And I'll actually use the same framework both in the first and second part of the talk, and it'll become clear how we apply it in these two settings. So in the HiC setting, what we have is not one matrix. We have, let's say, 3 matrices denoted by 1, 2, and 3. And for each of them, we solve this objective, which is denoted by the first term, and so we have T of them. So in this case, you have 3 of them. And in addition, we have this regularization term, where we are saying that depending upon how similar the different tasks are, we want to shift their low dimensional representations more close to each other compared to the ones that are more further apart. And then, so alpha is a regularization term, and then we can use that to control how much we want to weigh the reconstruction of the observed matrix versus how much we want to constrain the sharing of information. So that's the general TGIF framework, and there's a bioRxiv version of this, and if you're interested in the details, you can check it out. But TGIF can be used for both. So we have 2 versions of TGIF, one to study boundaries, which we call TGIF-DB, and the other one is TGIF-DC, which is for finding compartments. So what does TGIF-DB give you? So it gives you these factors or these low-rank representations, and using that, you can already kind of see the structure come out. But then with minimal post-processing of those factors, we can get out what are significant changes in the overall count, and that can give us what are boundaries. Then we can do statistical tests that help us to find what are differential boundaries. And I'll talk about a little bit more in subsequent slides. And then at the compartment level, here we take again at the whole matrix, at the whole chromosome level, we get the count matrix, the observed or expected. And here we can tune the K, which is a rank, and get both compartments and subcompartments. So compartments are basically the primary type of structure that was seen at the chromosomal level using single PCA-based type of frameworks. But NMF's rank can be used to find some subcompartments, and that can just come out from the low dimensional representations. So we applied both TGIF-DB and DC to different mammalian time courses. These are all differentiation time courses, but it doesn't have to be that. We can apply to any multi-sample type of a thing, which we have where there is information about how they are related. So now we want to be able to see whether doing this in a multitask setting is actually beneficial versus just finding tags and then doing a DE kind of or a D whatever test, differential test to see what changed. Now, as I said, this is a type of unsupervised learning problem. And we don't know what the ground truth is. We don't know what the true boundaries are or the tags are. But we know that there are other signals in the genome that can help us say what is a meaningful boundary versus not. And one of that is this architectural protein called CTCF, which has been shown to be enriched in many boundary regions. And so what we did was we asked for methods that have been used to identify tags and these pairwise methods, how do they look in terms of the CTCF enrichment? So here you see different methods. We have TGIF-DB and TAD compare, and we have these other methods which are generally taking the Hi-C matrix one at a time, and we are asking the boundaries that it found, how significantly enriched it is, for this particular time course data set. And what we see is that there are 2 methods that do quite well, and TGIF-DB is one of them, whereas the other methods don't do very well as far as the TAD enrichment, as well as CTCF is concerned. Now, the other thing that we can get from these types of methods is lower false positives. So to get to that question, what we did was we took a high-depth data set, and then we downsampled it, and we asked, do the tags that we get between the high-depth and the low-depth data set or across biological replicates, how similar do they look? And we measure that similarity based on a Jaccard coefficient, and we want a good, robust method that is not sensitive to depth and systematic noise, which should have a high score. And what we see is here, TGIF-DB and TAD compare is able to do much better compared to these other methods, and across also replicates. And then similarly at the compartment level, we can find compartments. And here again, to interpret this, we look at the enrichment of histone modifications. And we can find at k = 2, we can find compartments So we get the two compartments, the A and the B compartments, where one of them is enriched for these activating marks, whereas the other one has more of a tie between repressive and activating marks, and more on the repressive marks. And then we can also get subcompartments where we can increase the rank of the matrix and get more substructure. So we can identify these differences, and they seem to be reproducible across depth and across different replicates, but do they matter? How much do these changes matter? We have some anecdotal examples, but how do we ask that systematically? So what we did was we asked whether differential regions that can come from differential tags or differential boundaries or differential compartments, do they influence, are they associated with changes in gene expression? So I'm showing you here results for two of the data sets that we looked at. And what we see is it depends on how you look at how do you do this. So if you look at like a kind of a radius where you look at a differential boundary and you look at genes that are close by, we see there's a very strong enrichment of differentially expressed genes. So things that are associated with differential boundaries tend to be close to genes that are differentially expressed. We see that more at the level of boundaries rather than at the level of compartments, but also there is a signal at the level of compartments. And then finally, we looked at specific genes, and here's an example of a particular gene, the ESRG gene that Zhang et al., who originally generated the cardiomyocyte data set, found this to be associated with special types of retrotransposon types of elements. And this gene is highly expressed in the ES state and not expressed anywhere else. And we can see that it is associated with differential boundary. And we found this in another data set where it was not previously found. So it was kind of recapitulated where in both of them, there was a TAD boundary present. So that concludes towards the first, I'm coming to the first end of the first part of my talk, where I talked about finding changes in 3D genome organization by using a multitask non-negative matrix factorization framework. And we have two versions of it, DB and DC, where we can look at boundaries as well as differential compartments. And by applying these to mammalian data sets, we are finding these differences that we think can tell us something about changes in gene expression, but also helps us to reduce false positives compared to if you were to simply just apply one method and do the differential tests later on. So I'm going to shift gears and now tell you about gene regulatory networks. I don't know, should we do questions now or should we do it later? Any burning questions before I move to the next phase of my talk? Okay, I'll just move on then. So I'll now talk about gene regulatory networks. This is an area that we've been passionate about from the beginning when the lab started, and me before that. And so gene regulatory networks, some of you might know this, but just so we are on the same page, we think of gene regulatory networks as the control machinery in cells. These are key pieces of the machinery that are responsible for what genes get expressed when and where. And we think of them as networks because they are defining connections between what are called these regulatory proteins or transcription factors and other types of control proteins that regulate every gene. And we want to look at this from a genome scale. We want to know what are all of the regulators of a particular gene. But identifying these gene regulatory networks experimentally and computationally has been a challenge. The reason why it is a challenge is because gene regulation is complex and many levels of control, and it is there's not one network. It changes. It's fundamentally context specific, and we can't really map them all because it's, again, a type of unsupervised learning problem. But with now we are in a great time. We have this revolution in technology, which is single cell and now spatial and multi-omics, that gives us the ability to get these high dimensional readouts for individual cells across different cell types and contexts. And these data sets are becoming more and more available. And this opens up new opportunities towards understanding gene regulatory networks. Now let's maybe think about some of the key problems that we might want to address when we want to find these gene regulatory networks. And some of these are more relevant in the cell lineage or in a dynamic system, but different components of these different problems I'll talk about are important for looking at gene regulatory networks. The first question is, what are the cell types? So we want to find cell type specific or context specific gene regulatory networks from the single cell types of data sets. So that's one problem. The second, which may or may not be relevant, is how are they related? The third and fourth are what are the gene regulatory networks for each of these different cell types, and how do they change? So I'm going to talk about the two of these questions in today's talk. So let's look at the first question of what are the cell types. So this is a very common workflow that people use towards analyzing single-cell RNA-seq data sets to find, let's say you're interested in finding different cell types. So you start with an input matrix, which is a cell by gene matrix. This is a sparse matrix. And we do some dimensionality reduction, and then we do some clustering on the low dimensional space, and then we want to know what these clusters are. So how do we do that? We find maybe DE genes, or we have a database, we look at markers, and we try to annotate this. So it's a very common workflow for defining cell types. Now, single cell data sets are becoming more and more complex, as in now we have time courses, and we have data sets that are coming from developmental stages and different species. And we want to be able to mine all of these data sets to make a more comprehensive map of different cell types. So these are multi-sample types of data sets. So how do we, again, leverage this complex heterogeneity of data sets? So here again, we are using a framework from matrix factorization. In fact, non-negative matrix factorization, again, comes to our rescue towards giving us a way to look at these single-cell RNA-seq data sets. And so what you're seeing here is how NMF can just be applied. And so this is the low dimensional, these are low dimensional representations, the U and the V. Now we have an asymmetric matrix, which is cells. So one of them gives us a cell representation, one of them gives us a gene representation. And then the cell representation can give us, if you just look at it, you can actually do a pretty decent job of getting out their first version of clusters, but sometimes it can help to do some additional clustering, but oftentimes it's just fine to just look at the low dimensional representations. And then in addition, you can actually get the genes that go along with these factors. So those groupings of cells and genes come together, then you can use those genes that go with each of the cell factors to see what are the genes that are actually driving the different clusters. And you may have to do some extra regularizations to get a clean program, but at its first cut, in many situations that we've seen, actually does a pretty good job in terms of finding what are informative genes for a particular group of clusters. But we want to look at NMF from a multi-sample setting. So we have multiple single-cell RNA-seq data sets. And there have been several approaches that are based on NMF that have been used to try to work with multi-sample data. The first simple approach is to concatenate the matrix and apply a single NMF, and that's joint NMF. The other approach is one of the popular approaches is Liger. That is a very nice approach. And what it does is it gives you a per data set low dimensional representation, but also has an additional component, which is the shared low dimensional space that it tries to jointly optimize. And so then it shrinks this and then shrinks the data sets to this common space, and then uses that to try to integrate the data. But all of these approaches have been largely for flat data sets, as in we are assuming that the data sets are largely similarly related. But as I said, as we look at more and more complex samples, the heterogeneity increases, so we can't make that assumption that everybody's as equally related. So what we've done is basically taken the TGIF framework and applied it to single-cell RNA-seq with some small modifications, but really this is an asymmetric matrix. And we have largely applied that framework to look at now single-cell RNA-seq data sets. So how does that work? So we compared it to a bunch of different methods that exist. And we looked at it in different settings. So one is the PBMC data set, which comes from 2 different platforms. And we are assessing it based on clustering coefficient, how well the clustering works. And this is assessed by these metrics like silhouette index or DBI, how well does batch correction work, and how do we get these integrated clusters. And what we see is that the methods there is a range, but among the top methods, TGIF is there. And we've looked at more complex data sets where there are more complex heterogeneity relationships, and we find that TGIF is among the top methods. There's no universal winner, but overall, when we look at it, we do a decent job of getting pretty decent clustering and good batch correction. And we've applied this framework to different collaborator data sets, and I want to tell you about one of these data sets that that we are working on, and this is in the retina. So quick introduction into the human retina or the eye that helps us see. In the retina, so there are several cell types. Among the ones that enable us to see are called photoreceptors. And there are 2 main types of photoreceptors, the rod cells and the cone cells. And these are the cells, the cone cells are actually the ones that actually help us give this really nice, colorful vision. And these types of cells are determined to a great extent, are marked by special types of genes. And so there is a gene called NRL, which is known to be expressed in the rod cells. And then there are other genes that also similarly are marked with cone cells. And what happens is in many patient populations, there are mutations in some of these genes. For example, the NRL gene has been associated with a number of retinal diseases, and together with our collaborator clinicians, they have been using human organoids, retinal organoids, 3D organoids from human patient cell lines to study these different mutations and also compare that to wild type, and to try to see what are the programs, gene expression programs that can be used that differ between wild type and mutants, and try to see what if we can help them manipulate these different cell types to make them look more like the wild types. So this is a team, and so we have these organoids, and what we have is single cell RNA-seq data. But these are coming from different genetic lines, and they also come from different time points. So we have 2 time points, a 180-day and a 270-day, and then there are the wild types, and there are 2 types of mutations, NRL1, NRL2. NRL1 is a more severe mutation, NRL2 is a less severe mutation. And this is a workflow where we give these data sets to TMF, get clusters, and then we try to look at what are the gene programs, and I want to tell you a little bit about that. But first, we compared to the batch effect property here. And so there are 2 batches, A and B, and they come from 2 ages. And we first applied the simple approach, simple NMF, where we are doing something like joint NMF, where we concatenate the matrices and then seeing what the representations look like. And it does a pretty good job. Simple is beautiful. But what it does is it tends to be a little bit sensitive to the batch. So what we see is that these 2 where they come from 2 different age groups, but they are the same batch. It tends to look a little bit similar, so it tends to like do this. But we don't see that in the case of TMF. So TMF removes that, so that's nice to see. So now we can go and see what it is telling us about the biology. So here what we did was looked at the clusters and found genes that are driving these clusters, and we tested them for enrichment for different cell type markers. And we see different types of cells. We see photoreceptors, which are the rod cells and the cone cells, but we also see other types of cells. So basically these retinal organoids are quite diverse in terms of the different cell types. It captures a nice spectrum of different cell types. But the ones that we are more interested in are the photoreceptors, which constitute the majority of these different cell types. But we find, and we further look at it by presence of different marker genes, and we can see that there are 4 clusters, 6, 13, 15, and 16, that we call them as photoreceptors. And We are not sure whether they're actually rod or whether they're actually cone, but some of them have a tendency to be more like rods, some of them have a more tendency to be like cones. So we are looking at these clusters, and we are looking at this data. This is actually pretty new, brand new work, not published, trying to get it published, trying to actually write it up. So this is, these are, and so what we can see is how these actually distribute across our different lines. And what we see is that the photoreceptor, so cluster 6, which we are calling more rod-like, is nicely present in the wild types, but then in the mutants, it's actually gone, okay? But in the not so severe mutant, there's a tendency in the cone population. And then the other one that we see is, so I should actually clarify this. So what we did was we actually took the cells here and we tested them for enrichment of genes that we know are associated with what are called rod cells. And we see that, and rods and cones, and we see that rods are gone, but then some of these genes are actually, some of these cells actually have a cone signature. The programs of rods and cones are actually quite shared. So we see that cone signature come out in the less severe mutant. But then we also have a cone population that we see are all conserved across all of the wild types and the mutants. We can further dig deeper into these different gene programs, and we do that by looking at the low-dimensional space of the genes and then apply clustering. And here we did graph clustering with KNN graph, and we can get what are these, what I call as gene sets. These are sets of genes that exhibit similar low-dimensional representations. And we can see that, and then we test them for Go enrichment, and this is for 2 of the clusters, 6 and 13. And for 6, one of the gene programs is interesting, so gene set 1, where it's largely present in the wild type and really not that present in the others. And we see it's enriched for visual perception and so on and so forth. And in the cone, what we found was a particular cell cluster, number 2, which is this one. It is more associated with the early, like 180 time point, and it is enriched for developmental processes. So there could be something about these time points that these programs are actually capturing. We're still interpreting these results, but they look quite interesting. But so that's where we are. So again, this is another example of dimensionality reduction, clustering type of problem where we have used NMF in a multitask setting, and we have used now its low dimensional representations, both at the cell space as well as at the gene space, to find cell clusters and also find gene clusters. And they can tell us something informative about these complex systems. And in applications to retinal organoid data, we are finding patterns that help us discriminate between what could be wild type cell populations, photoreceptors versus mutant cell populations. All right. So, and now I'm going to finish up with gene regulatory networks and defining them and comparing them. And most of this is actually published, so I'll go quickly and we'll have time for questions. So here I'm interested in this question of what are the GRNs, how do they change? And for those of you who have not encountered this problem, here is a very simple intro to the GRN inference of the network inference problem. What this is basically, what we are saying is that we don't know what the network is, but what we have is measurements for the nodes from high dimensional matrices. And this was actually now 20 years or almost 20, 30 years ago when microarrays emerged, and there was pioneering work by Aviv and Daphne and Dana and all these people who used probabilistic models to infer these gene regulatory networks. But what we have is basically a data matrix where we have nodes and experimental conditions, and methods use variation or different army of methods are out there to try to infer what the underlying structure might be that explains the data well. And so this is by nature unsupervised. And what we have in its full form is a graph structure learning problem as well as a parameter learning problem. And we have methods for bulk data as well as single cell data, many reviews. For single cell in particular, there is a number of methods. We can group them into these broad categories. One of the big differences is how do these methods actually model an edge? What do they mean by an edge? And this can be information theoretic, it can be a probabilistic, it can be correlation. And so there is this whole family of methods. And then there's this notion of pseudo-time, which some of you might have encountered, that can be computationally inferred, and some of these methods also incorporate that. And then more recently, people are trying to extend these methods to go beyond gene expression, and there are some methods that are out there. And for single cell data, what we have is, again, we have this matrix, we have a cell. Here I have a gene by cell matrix, but a single cell RNA-seq data set is all you need to get you one reconstruct. So that's why it's so exciting now to work in this field, because the sample size problem for these big learning problems is now not there anymore. There are other problems that we have lots of samples, but we feed it to a network inference algorithm and out comes a network. And there have been nice reviews and probably more recent reviews and also benchmarking studies to ask what methods are actually good out there for these different problems. So I want to tell you a little bit about a benchmarking study that we did. This was a collaborative work by several members in my team, and you can read up more about it. I want to just touch upon the main points here. And here, what we did was we primarily focused on single-cell RNA-seq methods, and we compared different methods that came from different categories, and we assessed their performance based on F-score and area under the precision recall curve. So I said this is an unsupervised learning problem. How am I assessing this? So for some cell lines, for some biological contexts, we have so the gold standards are not perfect, so you should always take these with a grain of salt. And we don't really have lots and lots of like gold standards, like labeled data, but we have ChIP-seq experiments, we have knockout experiments, and we can use that to see how well did we recover those experimental data set based interactions. And this is looking at different cell lines. This was embryonic stem cell line, and then there was a dendritic cell. And for those cell types, we have some decent data sets that we can use to try to benchmark these data sets. So we looked at different methods here that you see, and then we also looked at Pearson's correlation, just as a very simple baseline. And surprisingly, we found that the methods that are developed for single cell do work. However, Pearson's correlation is also pretty good. That was interesting. There are some other things that we go more into the detail where you may not want to just use Pearson's. So the metrics actually matter. These are global metrics. You look at local metrics or go dig into some of these predictions, then these other methods are important. But other methods that are important and useful as CNIC, and a method that we developed, did pretty well, PIDC. And many of these results actually are consistent with Pratap, another benchmarking study that maybe people have encountered. And then, so we asked, what if we added more information? So one way, and people have done this in different ways, one way to do this is to estimate something called transcription factor activity that helps us to go just beyond gene expression and also integrate auxiliary sources of information where prior knowledge is trying to say, I have a regulator and a target gene, do we have evidence that they interact, and can we incorporate that to help the algorithm? And when we do that, we do see an advantage for several methods, and especially when we use this estimated TFA, we see that methods that didn't use that but have the flexibility to incorporate that also are benefited. Now, so adding prior information or adding other types of information can be beneficial for inferring gene regulatory networks. Now, there are still challenges, not only that they are using only single-cell RNA-seq data, there are challenges when we want to think about it in the context of a developmental system or things where cell types are related. And the challenge is that most methods don't actually account for the fact that these things are related. So they deal with it in a more of a single task setting. And what we want to do is not restrict ourselves to that limitation. So we developed an approach, SCMTNI, which takes in as input single-cell RNA-seq data sets for different cell types. And if you have also attack-seq data, single-cell attack-seq data for the different cell types, single-cell or bulk, or if you just have motifs, it takes both of these types of information, and then it and then it infers and a lineage structure, and it infers a gene regulatory network for each of these different cell clusters. So some of the key points, it's based on probabilistic graphical models. It's based on a type of model called dependency networks. And again, it uses multitask learning. But how does it do this is in a different, it does it differently than how I was talking about it in the previous parts of my talk. So the way we do this is by defining this particular objective. I'm just going to tell you very briefly. But we have, so what we're saying is you want to think of this within a Bayesian framework where we have this D, that is the observed, and we want to be able to estimate G and theta. I'll not talk much about theta, but G, so what is this? D is actually a collection of data sets. So we have M such data sets, and G is actually a collection of graphs, and we have M such graphs, and M is a number of cell clusters or cell types. And P of G is prior information. And so what we're saying is you're going to incorporate all this sharing information in the prior, this P of G. And this is how we write it. We make some strong assumptions of every edge is just independent. We make those strong assumptions to be able to compute this in a fast way. But this is, again, an area that people can explore. And there are not very many nice ways to do this. But again, from an algorithmic point of view, I think it's very interesting. But what we have is basically a pairwise prior that we then that we can use to try to incorporate how similar should the graphs be when they're coming from these different cell types. So so that's the general framework, and we first again benchmarked it by looking at simulated data. Here we generated graphs from a simulation where we knew the underlying ground truth, and we compared it to methods that incorporate multitask learning and methods that don't. And we find that generally methods that use multitask learning are beneficial compared to methods that don't. And we have a whole workflow where we have applied SEMTNI to different data sets, and I'm just going to give you a quick vignette of one of them. So these are some of the things. So, yeah, so this is general workflow. So what we do is, let's say that you have single-cell RNA-seq data and attack-seq data, and it's not a multi-ohm experiment, as in for a cell, you don't have both RNA and attack. You can still integrate it. We use Liger here. We get integrated clusters. Then we get a minimum spanning tree, and that's how we get a cell lineage structure, but you can use whatever you like. What we need are cell clusters and related by a tree. And then we compared it to different methods, and then we looked at dynamics, how the networks actually change. So we applied to different systems right now with our collaborators. I'm going to talk about the part that we published in the paper. And this is with my collaborator, Rupa Sridharan, who's studying a process called cellular reprogramming, where we go from these differentiated cell types to become pluripotent. And we have a single-cell RNA-seq data set and also a matched RNA attack-seq data set. And we integrated this with Liger, and what you're seeing here are UMaps, where the different clusters are here and the different samples are here. And these are the embryonic stem cells, and then the rest of it is basically a mix of, so it's a pretty mixed data set where, and these reprogramming populations tend to be very heterogeneous, and so which is what we see. And this is why you should use single-cell RNA sequencing. So the clusters are what actually capture the differences, not the time points or the days. But we see that RNA and attack actually nicely co-cluster, and that can give us priors for each of these different cell clusters. And so that's more clear over here. This is for a short kind of time course. And we can see, for example, cluster 4 is largely made up of these what we call as the MEF, the starting point, and then we have cluster 5 and cluster 2. Cluster 5 is what is associated more with the embryonic stem cell state. These are successfully reprogrammed cells, whereas the other endpoint is the cells that actually didn't reprogram. So one question is, how do these different cell clusters differ? And are there genes and GRNs that make them different? So we've looked at it, and without going into too much detail as to how exactly we found those differential subnetworks, I'm showing you these results. I'm happy to talk to people after that later on if you're interested. But what we find is that in the in the ESC cluster, we find these subnetworks that have these connections around these genes like ESRRB, which are known to be markers for the mouse embryonic stem cell state. And we see that there is, and that is not there in these other cell clusters that did not reprogram. Instead, the other hubs that could be in potentially as bottlenecks that could make these cells inefficient. And we look at this more broadly across the entire lineage, and we can find interesting regulators, and we find that the cells that is reprogram are the ones that have a strong embryonic stem cell program and regulators, whereas those that don't reprogram still have a persistent MEF program that still is there. So these are some regulators that just hang out, that just prevent these cells to reprogram. So that brings me to the last part of my talk, and that is, single cell is really has a lot of opportunities for machine learning, unsupervised learning, for many problems, and here especially for gene regulatory networks, and with the ability to get also multimodal data like accessibility, we can get to the cell type specific GRNs, which was very, very difficult before these types of methods. And we developed an approach that tries to incorporate these types of data sets to infer GRNs for different cell types and to try to leverage how they may be related. How they're related is something that we expect as input right now, but that's again another area that is of interest where you can think of another learning problem. But there are many methods that are out there to look at trajectories. But using our framework, then we can identify context-specific networks and then identify differences that we can then follow up with validation experiments to see how these GRNs actually changed. So that's pretty much it. So just back up at the gene regulation, multiple levels of control. All of these offer examples of unsupervised learning problems, and I talked about two of these. And if you're interested to know more, please, I'm here for the rest of quite a few days of the conference and happy to talk more. Want to acknowledge my group and my collaborators. Most of the work that I talked about is from Erica, Sunny Grace, Junha, and many of them have been doing all this. We've applied to plants also, so all of us are working on these problems. And if you're interested, I also am always looking for people. And I'm thankful to the funding agencies. And I want to just put one little thing, which is I also direct the PhD program in biomedical data science. If the students interested to pursue PhD or master's in biomedical data science, please also find me during the conference. With that, I'm happy to take questions.
This is Jundin from McGill. So first of all, thank you for the very comprehensive, inspiring talk about the GRN. So I have actually a quick questions. For the GRN inferences, right? Often the case, we, as you showed in your talk, the top preference performing method, some of them are, a significant portion of them are correlation based, right? But often the case we need GRN for interventions. Say, I lock out a TF, say how that influence the downstream gene expression. So to some extent, we're more interested in the causal ones, right? The causal GRNs. And then, so in that sense, inference of the causal GRNs often require sort of time series. You need to know what happens earlier and what happens later on, right? But in single cell studies, often the case you do not have a single cell time series, right? You may have only a snapshot. You have like healthy versus the disease. In that case, I want to hear about your ideas and about how we can usually deal with that situation scenario.
To infer causal GRNs?
Yes, without time series information.
Without time series information. But what we have now is Perturb-seq. and perturbation technologies that are becoming more and more common. So causal GRNs is extremely exciting, and there are actually, there is work out there, but it has, and that people are thinking about this. We are also like, working towards that, but there's not anything ready. But there are methods that actually try to get to those causal interactions, but not only from perturbation data. There are actually methods that try to get to that from observational data, but you can make some assumptions, and so there are some methods that are out there that have been used to. There's some classical methods that are available that you could use to find causal interactions, and it's still like, with the availability of perturb-seq data sets, these kinds of specialized methods are going to be these, more formal methods that actually think about a graph and a model are going to be very, very useful. Yeah, so I'm very much in, I will not use Pearson's correlation, even though it's reasonable, it's fast. I'll not use Pearson's correlation. I will use the method that, one, we like to develop methods because we apply them, because we understand how they work. These methods are hard, and what they tell you, you have to always take it with a grain of salt. So there is a lot of room to actually improve. But so with perturb-seq types of data sets, there is a lot of opportunity to try to infer GRNs, causal GRNs.
So a follow-up question on that. So I know perturb-seq will be very extremely useful, but it's very limited, as far as I know, right? It's only, for example, if I'm interested in one specific disease, maybe I can't find available perturb-seq for that specific scenario, right? What in that case, there's no way we can use it.
Okay, yeah. So there are methods that have been used, that can be used for with observational data, and there are methods that try to use time series to get you some of those. It's again an open question.
All right. Okay. Right.
And there have been, like, people are trying to model dynamics by looking at, like, splicing and that, velocity and things, but it's not there yet. So yeah, that's a good question.
All right. Thank you.
Hi, Sushmita. Very nice talk. So I think it's a great idea to leverage the tree structure in multitask learning. I may have missed this, but if you apply such methods to multi-species data, like single-cell RNA-seq or the HiTSeq data, do you need data for ancestral species? And if yes, how do you get that?
You don't need it for ancestral species, because we are not actually observing anything. We are just, we can reconstruct it. Yeah, we don't need it. Yeah, we don't need it.
Okay, thanks.
But there's another problem with multi-species, actually, and that's the mapping problem, which I can get into later on. It's a very interesting problem.